Hi guys, my name is Randy Holloman. Today we're going to get after it by looking in the research article titled Complex Genetic Determinism of Male Fertility Restoration, specifically in physa acuta, which is a snail species that lives in an aquatic environment. Um, the species is significant because it has a gynodesis system sexually. Basically, what this means is that it has female only producing organisms and both female and male producing organisms known as hermaphrodites within it. Similar to some plants, um, it's largely unstudied in animals. It's much studied in plants, and basically what the whole point of this article was to study the cytoplasmic male sterility and nuclear restorer genes and how they act upon this population. Um, Basically, they wanted to get a big understanding of the evolution of sexual systems and the persistence of um, the ability to produce male fertile organisms and female only organisms. So, so their hypothesis for testing within the article, basically done by the researchers, was male fertility restoration in physa acuta is genetically complex. Um, they also wanted to know if it was just acted upon by a singular nuclear gene, um, and this right there, that hypothesis was followed by the results, um, as seen below. Male fertility restoration is not controlled by a single Mendelian gene. Instead, it's acted upon multiple complex genes. And as we see in the data graph right here, we can see that there's a lot of variability here. If it was controlled by a single gene, it would have 50-50 ratios or 100 to 0 ratios or 75-25 ratios. But there's a lot of variance. There's also this semi-sterile organism that enters the population, and that's controlled by weak modifier genes that just allow it to kind of exist, but in a weaker version. Um, but then the strong dominant restore alleles allow the fertile organisms to exist and basically restore the population's fertility. So they also found within the study that environmental factors could shift sexual phenotype expression. And basically what that means here is that there are multiple factors such as male-to-male competition or just a male not being selected on that carries this fertility gene or genes, um, and they could not be selected on, and it could just not enter the population, and sterility could continue until a male is acted upon that has these genes. So, the significance of the findings here was that it shows the complexity of evolutionary genetics and male sterility and Physa acuta, and it suggests common evolutionary forces across all areas of the population. Um, It shows that sexual system evolution is affected by interactions of cytoplasmic and nuclear genes, basically getting back to the point of a very complex system. Um, it suggests environment and factors such as competition can affect how restoration appears. Basically, what the researchers talked about was how they want to study in the future, and they want to build on this research by seeing how it interacts with an environment. And competition of population, and it shows the need to find the genetic regions for fertility restoration to determine how gynodecy is maintained. And they just want to know in the future how it's it coexists even more within the population. And now, just one final thought. Uh, Male fertility restoration is complex in genetic layers. It's very intricate. The gynodecy evolutionary maintenance is complex. It's there's a lot of factors that go into it. So I just want to say thank you.